Bio30, welcome to the second video on protein synthesis. This is all about drawing the picture. So more or less listen to what I'm saying, watch what I'm doing, and I will tell you when to draw your picture. So yesterday I asked you guys to do some quick practice questions just at the end of the lesson in your notes. And I just wanted to quickly go over those. So you had a set of DNA and it looked a little something like that. I hope I copied it right. And I was asking you guys to determine the amino acids produced. So you could have gone about this two ways. You could have gone DNA, mRNA, tRNA, then amino acids. I skipped the tRNA and I went straight to amino acids because reminding yourselves, mRNA codes for amino acids. So first I found my mRNA. It's just the complementary strand to DNA. So T matches with A's match with U's. C's and G's are the same. So I went through and I made all my different codons, my triplets. So there's AUG, GGC, CGU, and I went through and I made all of my different mRNA. I then went over to my amino acids chart and I started to find which ones coded directly for each one. So I got the start codon here. Some of you may have written down MET, methyl-iodine, but reminding yourselves if it's at the very beginning, it's a start codon. Then I have glycine, arginine, serine, tyrosine, and arginine again, and then stop. In the process of making these, you can just use the abbreviated forms, and I also don't expect you to know their names. Just using their abbreviations is perfect. So that was question one. The second question asked you to take the mRNA given to you and find the associated tRNA. So mRNA is essentially complementary strand. So again, U's pairs with A's pairs with U's, C's and G's, right? So UUU becomes AAA, AGG becomes UCC, and so forth. So those were just what they looked like there. And then last but not least, it posed a question to you is what organelle plays a role in protein packaging? And that is the Golgi apparatus. So quickly look over your answers, make a note to yourself if you didn't get them right to ask me a question and figure out where you went wrong. Because this here, ladies and gentlemen, is now the rest of our chapter, is going from mRNA to amino acids. So as promised, I'm going to show you a picture of what the process of transcription and translation look like. So you have a little space in your note package that says draw a diagram of protein synthesis and write the steps involved in transcription and translation. Writing the steps down is going to be key, but the pictures are hard to make picture by picture, so I'll let you know when to draw a picture. At this point, you don't need to write anything down unless you want to remind yourself that the nucleus, is where transcription happens, and translation happens in the cytoplasm. And that's what you're seeing in my picture right now. So step one is protein synthesis is transcription. So we're making DNA into mRNA, and this happens in the nucleus. So writing down step one somewhere on your sheet is key. Do you need to draw this? No, because it's going to be too crowded inside your nucleus. You can try to if you'd like to, but you do not have to. So step one is RNA polymerase unwinds and unzips the DNA, much like helicase did, except this time we're using RNA polymerase. So if you're looking at your sheet, you may have something drawn like this in there if you want, but you don't have to. I would wait till the end. So there we go. RNA polymerase has unwound our DNA. There's our DNA. So let's zoom back into it. So in the second step, so step two that you're going to write down, RNA polymerase adds nucleotides of RNA to the DNA. So A matches with U's, and RNA should look almost exactly the same as the non-coding strand, except T's are now U's. So if this is my coding strand at the bottom and my non-coding strand at the top, when I add my RNA in, using my RNA polymerase, you'll notice that it should look identical to the one up above it, except where there's T's, there's now U's. So the orange color on my sheet is my RNA. So messenger RNA is now made, and that is what my picture looks like in my notes. So I would draw that down as a way to remind yourself of what's happening during transcription. So the next step is taking this mRNA and putting it into the ribosome to make some proteins. All right. So we are going to enter the mRNA into the ribosome, and it's going to look something like that. So this is step one of translation. Now that my mRNA is in there, I need to use tRNA, my invitation to the party to bring the right amino acid to the ribosome. So that's step 2. So what you're seeing here is the structure of tRNA, that's the blue piece, and attached at the light bottom, the light blue piece, is the complementary tRNA. So it matches up like this. On the top, you will notice a blue circle that says the word start in it. Start matches AUG, because remember, I am making my amino acids using my messenger RNA. So AUG means methylamine, or in this case, start, because it's the first codon. So tRNA brings that start codon to the ribosome and attaches it on the outside. Step 3 is that this process continues on. So here comes another tRNA. There it is, matching it with its mRNA. And this time, mRNA is coding for arginine, ARG. The amino acids are being added to the top of the tRNAs to build a protein chain. So what you're seeing here is that tRNA will release it and we will start creating a protein chain. We are going to continue steps 2 through 4 until we reach the stop codon. So step 5, here we go. Adding asparagine, it's going to link in and we're going to move down. And now we have our stop codon. And now we have an amino acid chain. This chain is now going to become a protein. So it's going to be sent to the Golgi to be packaged up. And that's the lovely squiggly lines inside your cell there. So ladies and gentlemen, that is the process of transcription and translation in a drawing. I went through it really fast, so please go back, make sure you add all 5 steps for translation. Make sure you add the 2 steps for transcription. Your picture in the end should look a little something like this. If you want to keep tRNA in there, like I did in my picture, this is actually what my picture looks like to remind myself that tRNA comes in and drops off an amino acid, you can. So again, have your picture ready in there so you know what you're drawing. The steps are written down, and we will be doing some practice today in the process of transcription translation. So when you are done drawing, underneath it says a translation exercise. What I want you to do is go through questions 1 through 4 and make mRNA codons from the DNA I gave you. Then I want you to try the protein synthesis practice, which essentially gives you the non-coding strand, the coding strand, mRNA, tRNA, and amino acids. And I want you to fill in this chart trying to figure out what the amino acids are. There are then 8 questions after that I want you to answer. I know this seems like a lot, but this is all practice. From there is, again, moving backwards in our understanding, some DNA replication questions. So at the bottom are gene mutations effect on protein exercises. So now we're linking the idea, ladies and gentlemen, of gene mutations, protein and replication, so we're talking point mutations, with what happens to the protein. So in this time, you're going to code for the original amino acid sequence by using mRNA. Then you are going to go through and do all the substitutions, additions, and deletions. I will post answer keys to all of this later today. Have a great one, and I'll talk to you later.